Alrighty, just give it one more minute before we get started. But thank you all for spending your time today to listen to Dr. Narvaez talk about her work and being a member of or part of the TIC IPM working group. My name is Zach. I am a coordinator with the IPM Institute, and I'll be just giving a little background in the Institute before we let Zoe get started. Alrighty, so who we are. The IPM Institute was founded in 1998 with the goal of increasing adoption of integrated pest management in agriculture and communities by using an educational and solutions-based approach. The organization is split into two groups: the Sustainable Food Group, which focuses on food supply chain sustainability, and in many cases work directly with growers, and the group that I work in, Sustainable Communities, focuses on sustainability education and resource development as it relates to people, pets, and pollinators in community spaces such as parks, fields, schoolyards, and buildings. Going in one step further, we have a few different programs that I'm a part of, including Green Shield Certified, a program with the mission to reduce use of pesticides and provide best use practices for pest management professionals, facilities, and landscape companies. The Pest Defense for Healthy Schools, has a focus on educating K through 12 staff about IPM and best practices to limit exposure and impacts to students, with free courses on the website you see there. And then, as you're all aware of it, because you're here in this meeting, the TIC IPM working group, with a focus on integrated tick management and education around ticks and tick-borne diseases. We have the virtual Tick Academy, which will be hosted again this year in October. And we also produce some educational resources through the North Central IPM Center, in which here we have technology their funding. We got funded again in June, so I want to thank them for their contributions. And just Tick Academy updates, we are still waiting on getting two more speakers for our second day of the Tick Academy. We're looking specifically for veterinarians and medical clinicians to talk about their experiences and their work with tick and tick-borne diseases. So if you have any recommendations, please feel free to reach out to me. And with that, I will hand it over to Zoe just after I launch this pre-presentation webinar question. The question is, what are the most likely country of origins of the Asian longhorn tick? And this is multiple choice, so you can select multiple options. I'll put this up for a minute and then all yours.
Is it just for the chat? Is it like really echoey when I'm talking? Is my audio okay? Okay, thank you. Okay, I'll go
Alrighty,
ahead.
let's get a minute. All yours.
Okay. Hmm. Let me just get the speaker screen out of the middle of my screen. Stop. Okay. Good. Okay, yeah, I'll get started. Thanks everyone for logging on and being here. My name's Zoe. I am currently a postdoctoral researcher at the Midwest Center of Excellence for Vector-Borne Disease. This spring, though, I finished up my Ph.D. in entomology at Rutgers University, working with Dr. Dana Price and a bunch of other really terrific people on the East Coast. While I was there, I got to contribute to some very diverse tick-related projects, which was really great for me because I got to learn about a bunch of different species and pathogens and techniques that are really relevant today. That being said, the title of this talk I know is very broad and not super informative. So I'm going to give a quick rundown of the topics that I'm going to cover. So I'm actually going to be talking about 4 separate tick species today, all of them undergoing really interesting population dynamics in the northeastern US and beyond. First up, I will be covering some work we did with the black-legged tick, Ixodes scapularis, in the urban forest of New Jersey, then talking pretty briefly about a cryptic, but I think really interesting species called Ixodes kirinsi. Then we'll be zooming out to a more global scale to talk about the Asian longhorn tick, which we all know is on the move in the USA in particular. And then if we have time, I wanted to share some of my non-dissertation work that I really enjoyed still with the Lone Star tick and its associated pathogens, specifically Ehrlichia pathogens. So first up, we do have Ixodes scapularis, black-legged tick, and I'm just going to dive right into that background that you have all heard before. Just to quickly emphasize that with very limited exception, black-legged tick-borne pathogens like Borrelia burgdorferi, the agent of Lyme disease, or human infective Anaplasma phagocytophilum, they're acquired by a tick during an early stage blood meal when they feed on an animal reservoir of disease. So the classic example, I hope you can see my mouse, it's of course that white-footed mouse, which we know can maintain several agents of human disease. In this scenario, the tick picks up microbes from that reservoir and then maintains the infection into the nymphal stage, at which point it's more likely to bite something larger like a human and pass on that pathogen. It then becomes an adult, and if it's lucky, it'll feed on something larger like a deer, which won't acquire the infection, but which will provide the tick with a large enough blood meal so that it can produce eggs and start the cycle all over again. So the bottom line here is that this tick in particular was dangerous to humans because of what it fed on previously and because that animal was a reservoir of disease. Fortunately for us, this is not always how it happens in nature. So in a healthy forest ecosystem, black-legged ticks might feed on a variety of hosts, relatively few of which should be carrying human pathogens. However, this can start to change when our forests are fragmented by human development. There's a lot of complexity and ongoing discourse to this, but it's generally accepted that disturbed forests tend to suffer a loss of biodiversity, which is usually seen first in the decline of large predator populations, and then in the proliferation of species that we call urban adapters. Unfortunately, these urban adapter species are thought to include the white-footed mouse, which we know is a reservoir for several human pathogens, a very important one, and also the white-tailed deer, which supports really large populations of black-legged ticks and other species. So by this theory, when we fragment our forests, we're ending up with smaller patches that might be carrying a disproportionate risk of tick-borne illness. However, this hypothetical effect has been pretty difficult to demonstrate in nature, and the results from the Northeast in particular have been pretty mixed so far. Notably, no one has looked into this potential association in New Jersey, or at least they hadn't a few years ago. But we actually thought this might be a really ideal place to approach this question on a larger scale. So New Jersey, if you know anything about it, it's notorious as the most densely populated state in the US. But pretty incredibly, it's also maintained the highest density of preserved parkland in the contiguous US. We are second only to Alaska, I believe. The result is that we have lots of not only large swaths of state forest land like the Pine Barrens and the Appalachian region in the north, but we also have lots of smaller forest patches that are surrounded by human development and chances for human contact with tick habitat ultimately. So to take advantage of this unique environment, we designed a study aimed at assessing the relative risk of black-legged tick-borne illness to visitors of urban forests in New Jersey. And when I say relative, I mean relative to much larger contiguous rural forests. So our study sites are ultimately compared using a bunch of other variables, but for the sake of just this presentation and summary, I want to focus on that comparison of urban versus rural forests. And I'll explain how we define those. So we have 30 study sites total, 15 of these being very large rural forests of minimum 500 hectares, most of them much larger than that and kind of contiguous with other forested regions. And then the 15 other sites were considered highly fragmented urban forests of maximum 180 hectares, but most of them much, much smaller than that. And all of those urban sites were entirely surrounded by medium to high density human development. So as you can see, on this side of your screen, if my mouse is even showing up, there is an aerial example of one of our urban forests. You can see it's really entirely surrounded by high density human residences. It's up in Bergen County. And then on the other side is our statewide distribution of urban and rural forests, which we tried to make relatively homogeneous as much as we could. So we surveyed these sites throughout 2022 and 2023, visiting each site 3 times per year. On each site visit, we would spend 3 man-hours collecting ticks using the dragging method, which you're all familiar with. It looks like this. During each visit, we also assessed tick abundance along a fixed transect, determined to be representative of that site, and we measured temperature and relative humidity and characterized or ground truth site attributes like forest type and shrub cover. After each visit, ticks were transported back to the lab. They were identified to species and life stage to sort out any non-black-legged ticks or lookalikes, and nymphal and adult black-legged ticks were thoroughly surface sterilized. We could then extract their nucleic acids and then run qPCR or RT-qPCR to detect the presence of our molecular pathogenic targets. And here's an overview of those targets representing the black-legged tick-borne pathogens that we currently know to be found in New Jersey. So we have, of course, up here, Borrelia burgdorferi, the famous agent of Lyme disease. We also tested for Borrelia miyamotoi, the rarer and lesser-known agent of hard tick relapsing fever. We tested for Anaplasma phagocytophilum, the bacterium that causes anaplasmosis. We screened for both variants, known variants of this pathogen, and also Babesia microti, which is the protozoan parasite that causes babesiosis. We would also, in a separate RT-qPCR assay, test for Powassan virus, specifically Powassan virus lineage 2, deer tick virus, which in my opinion, despite being the rarest, is pretty much the scariest of these pathogens because it has a very short transmission time, as little as 15 minutes, and there's really not a lot we can do to prevent or really do anything about illness once that transmission has occurred. So jumping right into results, this is an overview of our tick testing pathogen prevalence results. These are broken up by year and by life stage. And again, what I'm showing for this presentation is just graphs of urban versus rural comparisons. So first up, we have the nymphal ticks from 2022. This is one cohort of ticks. We collected around 600 of these and found nearly 27% of them to be infected with at least one pathogen. You can notice from the scale of the y-axis here, get an idea of how many ticks are actually infected. Just note that this is not 100%, it's 30%. And these bars here, for each of the pathogens, the solid bar represents rural site infection prevalence, and that striped bar represents our urban infection prevalence. So the most prevalent of all of our pathogens was, of course, unsurprisingly, Borrelia burgdorferi, the agent of Lyme disease, over here in orange. And these two bars represent, like I said, rural and urban Lyme agent prevalence. You can immediately see there were far more urban ticks infected with Borrelia burgdorferi. This was significant by a chi-square comparison. We did see a similar discrepancy just visually for Anaplasma, but the sample size here was smaller, the difference not quite significant. For the remaining 3 pathogens, also less prevalent than Borrelia burgdorferi, not a substantial difference between urban and rural prevalence. I should note that the 3 Powassan virus positives shown here came from just 2 sites in the northwestern corner of New Jersey, which was really not surprising, as this is a very highly focalized pathogen that has only has confirmed cases in the northwestern corner of New Jersey, pretty much. So here are the adults from the same year. These are the nymphs, but they have had one more blood meal, so they are generally more infected with everything. Again, the Lyme pathogen was very prevalent and significantly elevated among ticks from urban settings compared to rural settings. We found it in just about 50% of our urban adults, as you can see from the y-axis. And the anaplasma results also significant for this cohort, following a similar pattern of high urban prevalence. Again, this was not maintained for the remaining pathogens. I will mention that our adult Powassan positives here were again collected from just a handful of sites. This was not surprising. What was surprising is that this time the majority of those sites were actually from southern New Jersey, where cases of Powassan are not known at all. So this was unexpected and slightly concerning, given that people and physicians in that region may not be aware that they are at risk locally. On that note, moving on to 2023, when we repeated this all again. So here are the nymphs from that year. This time, they're only slightly more infected with the Lyme pathogen at urban sites, not to the point of significance. And then we had very little signal for any of the remaining pathogens. But those patterns kind of reappeared when we tested adults from that year. Borrelia burgdorferi, again, elevated at urban sites. Anaplasma, more prevalent at urban sites. And the other pathogens quite rare and non-significant. For this cohort, we didn't detect any powassan, which was a good thing. Now, I have to give a quick caveat to the anaplasma results in particular. I mentioned briefly that in the US, we have 2 known variants of Anaplasma phagocytophilum, and only one of them is human infective. It's called the human active variant. These 2 variants can, though, be very difficult to distinguish molecularly. So in our detection assay, we do screen for both of them. And then we attempt to genotype after the fact, though we are not able to successfully do that for all of our samples. So basically, the previous slides showed all the ticks that were positive for either variant of Anaplasma. But here, I just wanted to break it down and show those samples that I was able to genotype conclusively as the human infective variant, which is more representative, of course, of human risk. So you can see that for most tick cohorts, nymphs and adults in 2022 and adults in 2023, the urban prevalence of the human infective variant was a lot higher than that of the rural prevalence. We did not see that disparity in 2023 nymphs, which was interesting because this was also the only cohort that didn't have an elevated rate of Borrelia-Burgdorferi in our urban ticks. Both of these pathogens are reservoired in rodents like the white-footed mouse, so it is quite possible that both of these 2023 nymphal dips are linked to the same fluctuation in urban rodent populations, which is pretty interesting. So as I mentioned, we also briefly assessed tick abundance at each site. Rural averages are, again, in solid gray. Urban averages are in stripes. And by a rank sum test, we had significantly higher tick abundance at urban sites for almost every year life stage combination, with the exception of adults in 2022. Now, in interpreting this, we should keep in mind that our density transects, they are really short. Definitely not sufficient to assess tick density at any one of our individual sites, but the fact that we saw repeatedly these cumulatively elevated densities at urban sites does suggest a real disparity here in terms of tick abundance or density. And it emphasizes that the ecology of these fragmented forests might be pretty conducive to the species of tick. Sorry, wanted to also mention, oh wait, no, I didn't. So anyway, takeaways based on what I've covered here. We can say in New Jersey, we are lucky to have access to so many forests so close to where we live and work. We may need to take extra precautions when we enjoy them because they have this potentially high prevalence of human pathogens and abundance of ticks. Lyme is, of course, the most prevalent tick-borne illness in New Jersey and in the nation. And anaplasmosis is actually really quickly gaining incidence. We wonder if maybe perhaps that could be in part due to intensification of risk in our urban forests, which are really growing. I should also mention that powassan virus, while nowhere near as prevalent, does represent a very serious risk to human health and one that might warrant some changes to our current public health messaging. People should be aware of this pathogen, for instance, statewide in New Jersey, not just in the north where cases have historically occurred. And given that really short transmission time of as little as 15 minutes, like I said, we need to place an emphasis on personal protection that will allow us to avoid tick bites in the first place, rather than removing them once they have already attached and it might be too late. Now, I want to also mention that forests are a lot more complex than their size alone, which is what I talked about here. And we did create models comparing them on the basis of several other variables. I do not have time to go into depth about that here, but if anyone's interested in the composition of New Jersey's forests, I would be happy to talk about that and show some data later. So here's another interesting takeaway from that project. We were focused on comparing black-legged ticks, of course, between urban and rural habitats. But in the process of collecting them, we encountered quite a few other species across the state. We had 8 total by the end. And black-legged ticks, unfortunately, were not even the most abundant. I say unfortunately because those are the ones I was looking for. There were instead a lot of Lone Star ticks, which I will hopefully get into later. But for now, I want to briefly talk about the least abundant tick we found, which was Ixodes kieranzi. So when I collected ticks, I would take them back to the lab and I would look at all the black-legged ticks or Ixodes ticks under a microscope just to be sure I was only testing the species of interest and no lookalikes. We do have other Ixodes confirmed in New Jersey. And in the summer of 2023, I was looking through a pool of ticks from Camden County in the southern, southeastern, western part of the state. I noticed that one of the nymphs was a little bit off right here. And there was also an adult female in the middle of the summer, which isn't something we see very, very frequently. And when I looked at them next to Ixodes scapularis, the black-legged ticks, there were some subtle differences. Most of them are not visible in these pictures, but you can see that our mystery ticks were a little bit rounder and less elongate, and also had these very angular mouth parts, which is what initially caught my attention. So I took them over to Andrea Jeezy's office because she always knows what to do with ticks, and she did give me some keys to look through. We used several keys for both the nymph and adult, but pretty weirdly, these ticks kept keying out to Ixodes affinis, which is a species only found in the southeastern US and never as far north as New Jersey. So this would be unexpected. But to know for sure, we sequenced the COX-1 and 16S mitochondrial genes, widely used to discriminate species. And sure enough, they were genetically Ixodes affinis. You might notice I have been saying both Ixodes affinis and Ixodes karensi, and that's because this species was renamed from Ixodes affinis to Ixodes karensi shortly after we found it. So when we published, we had to pivot to the new name, karensi, which was cool because it was named in honor of Dr. James Karen's, who created several of the keys that we use to identify this species. And in preparing this publication, we actually found that our detection was pretty much in line with the recent trend of northward range expansion for this tick. So karensi was historically found only in the deep southeastern United States, here in this historical range blue. But this started to change in the 21st century when there was a string of progressively northward detections. In fact, it was actually picked up in Delaware just a few months before we found it in New Jersey. And our colleague there, Ashley Kennedy, generously sent us a specimen to sequence along with our own. We, of course, expected all three of these ticks, 2 from New Jersey and 1 from right south of the border, to be really similar, if not identical genetically. But this was not the case at either of the loci we sequenced. So here we have a maximum likelihood tree of the 16S region, for which there are quite a few sequences available, thanks to a previous publication that found there to be certain haplotypes or sequence types that are dominant in the historical range, and others, distinct haplotypes that are dominant in the expanded mid-Atlantic range. So our New Jersey adult was most closely related to those northern ticks, which makes sense geographically, but the nymph here actually aligned much closer with the historical southern clade. And the Delaware tick, while closer to the northern group, did not fall into this clade perfectly. So it appears we may actually have 3 ticks here that arrive from separate populations, which suggests substantial gene flow from different locations across the seaboard. These ticks are known to feed on several species of migratory bird, which might explain how they are getting here in the first place. But according to the timeline we kind of have, their actual establishment may have become possible only recently, perhaps in part due to the milder winters we've been experiencing. And we care about this not because these ticks bite humans, because they actually do not, but this does not mean they are harmless. There's emerging evidence from the historical range that shows that karensi are instrumental in the enzootic maintenance of Borrelia burgdorferi and other closely related Borrelia pathogens. So in other words, they're passing these pathogens between their natural reservoirs, like mice. And this could theoretically impact the risk of Lyme disease when they move up into the northern part of the country, where we have Ixodes scapularis to bridge these pathogens from animal reservoirs to humans. So again, at this point, we know very little about these new neighbors we're experiencing in New Jersey, but they may be an important species for us and nearby areas to watch out for and study as their populations grow. So now back to that table of species. You may notice that the third most common tick we found was the Asian longhorn tick, Hemophysalis longicornis. If you're familiar with this tick, which you probably are, you may know that they were first detected in the US in New Jersey, and they love it in New Jersey. They are doing fantastic. The next project I'm going to talk about has to do with their greater movements, though, across the US and the world. This was a project that was introduced to me by my advisor, Dana Price, pretty early on in my PhD, and it did take me pretty close to the full 5 years to carry it out and get it published. But part of that was because I found it super interesting to dive into and spend time on, and also really relevant to the position we find ourselves in with this constantly expanding tick. So here's some background, although you will have heard it all before. This species is native to East Asia, historically China, Japan, and Korea, and more recently invasive of parts of Oceania like Australia and New Zealand. In this range in the Eastern Hemisphere, it's pretty well known as an agricultural, especially also veterinary and medical threat. It has a strong preference for certain hosts like cattle and dogs, but it's opportunistic enough to bite humans and other animals. I know I found all 3 life stages biting me at some time or another. And these tendencies allow it to transmit some zoonotic pathogens to us as well as to our livestock and companion animals. An interesting defining feature of these ticks is that overseas they have both bisexual and parthenogenetic populations. So this means that some populations have both males and females that will reproduce sexually, and others have only female ticks that can reproduce asexually. So basically, a single female can produce a few thousand eggs without even mating, which, as you can imagine, makes them especially prone to successful establishment in new areas. And of course, they also have that tendency to attach to domesticated animals that do a lot of traveling, aided by us. And that can really contribute to that risk of invasion. And of course, they did find their way to the US. They were first detected in Hunterdon County, New Jersey, which is where I grew up. They were found infesting sheep in 2017. Following this discovery, archived specimens from West Virginia in 2010 and elsewhere in New Jersey in 2013 were retroactively identified, which raises the question of how long these ticks might have actually been here before we noticed them. They have now been identified in, I believe, 21, it could be more, US states across the eastern side of the country, as far west now as Oklahoma. And just a caveat, according to climate modeling, they could establish successfully across much of this half of the country, all the way up into parts of Canada, and even do really well in pockets on the West Coast if they are able to get there. So given this developing situation, there are a few questions we're motivated to answer. First, we want to know, where did these ticks come from? Their range in Asia and Oceania, as you can see here, is pretty broad and includes plenty of different countries that we do lots of trade with, like China, or even, for instance, Australia and New Zealand, which do send us livestock. This brings us to ask, how did they get here? If they traveled on an animal or animals, was it a livestock shipment? If so, we may need to reevaluate our current inspection and quarantine procedures. And how many times were they introduced? This is actually pretty important because we want to know whether this was a single incident or whether they're being brought in repeatedly through a route that could still be open today. And if that's the case, we need to nip it in the bud to prevent future introductions, which could, for instance, include pathogens found in certain parts of their native range. Fortunately, there was a very thorough investigation into these questions published in 2020, led by Dr. Andrea Giese. The authors accumulated samples both from across the US range at the time and from overseas, and they sequenced a portion of the cytochrome oxidase 1 or COX-1 mitochondrial gene. This research showed that there are 3 unique COX-1 haplotypes or sequences found in the US. They refer to them as H1, H2, and H3. And basically, this suggests the introduction of 3 separate lineages of tick to the country. These authors also showed that all US ticks do come from parthenogenetic populations, which makes sense, and means that theoretically each lineage could have had a single founder. So the study that I'm going to talk about today is one that builds off of that foundational work by taking select samples and sequencing the entire mitochondrial genome, not just the COX-1 region. The idea here is that with this expanded sequencing, we'll get a lot more genetic information, allowing us to hopefully see some of the more fine-scale relationships between US ticks and ticks from specific populations overseas. We should also hopefully be able to see what kind of diversity there is in US populations, which will tell us more about how many lineages may be present. To get there, we first had to select geographically diverse samples, both banked from previous studies and sourced from areas of newer expansion, particularly in the US. New samples had their DNA extracted, and then with this template, we performed PCR to amplify DNA from the entire circular mitochondrial genome, which was a lot more difficult than I expected. It took the bulk of my time. It was done using 6 primer pairs published by collaborators in China that overlap to cover the whole genome with these amplicon lengths of 3 to 6 kb, depending on which combinations of primer pairs I use to Frankenstein each mitochondrion together. Once that was done, we would sequence the amplicons on our MiSeq system, clean up the output, map it to a reference genome, and make an alignment of the consensus sequences. At this point, we were lucky to be able to add in a bunch of extra sequences that were contributed by collaborators in China and Japan to really increase our representation of overseas genomes. And finally, once we had all of these mitochondrial genomes, we could build this great big maximum likelihood tree to evaluate the phylogenetic relationships. So here's the resulting tree. It is a cladogram with branch lengths off. Do not worry about squinting at it at all because I am going to zoom into each clade on the tree. But first, I want to use this very broad view to point out that all of our US samples and most of the foreign samples fell into these three COX-1 defined lineages that I discussed earlier. So H1, H2, and H3. And as we'll see more clearly in just a moment, within each lineage that we have these US ticks that are clustering in with Asian ticks, which confirms that all three haplotypes do represent foreign introductions rather than branching off of one another after arrival in the US. So now we are going to look into each lineage for relationships that might point towards introduction events. This is pretty speculative stuff, so you have to take it all with a grain of salt, but I think it's really interesting. First, we have H1, like I mentioned, a COX-1 haplotype that has largely been seen in the northeastern US, save for one sample we had in Virginia. Something that really stands out about this clade is that all of the US specimens we sequenced, shown in this red box, they're completely identical or clonal throughout the mitogenome. So this points towards a single introduction, most likely to the northeastern part of the country where this lineage is geographically focused. Now, the previous investigation, this haplotype was found to be really strongly associated with China. In fact, all of the H1 ticks found overseas were from China. So I assume this lineage most likely came to the US from there before I started this work. But pretty interestingly, our dataset also included this sample from Jeju, South Korea, which is an island off the southern coast of Korea. And as you can see here, this was actually the sample that was most closely related to the ticks from the US. Now, I mentioned the previous investigation did not find any H1 in South Korea, but they still suspected that this might be a source of introduction because these ticks really love to bite dogs, and the US does have several organizations and initiatives that rescue large groups of dogs from Korean meat farms, many of which are located on Jeju Island, and bring them into ports, including New York and New Jersey, for rehoming, which is really great work. But dogs on these farms, unfortunately, may be kept in really poor parasite-infested outdoor conditions, and as companion animals, they historically have not been subject to the same strict regulations and inspections that livestock, for instance, go through when entering the US. Now, I want to point out a potential caveat here is that we have only that one tick supporting this connection between Korea and the US, and Jeju is a noted stopover island for migratory birds. So it is circumstantially possible that this is a one-off specimen that does not represent the established H1 population there. However, this tick from Korea was substantially diverged from all the other Chinese specimens we looked at. So here is a principal component analysis, which looks really busy, but it can be boiled down to this spatial representation of genetic diversity. And this shows us how the Korea specimen here in green lies outside of this space, which represents the average specimen from China. And even further from that China space, we have the US specimens, represented by the yellow dot. So basically, we have evidence here to support that, unexpectedly, for me at least, this lineage most likely did not come from China, despite there being a very substantial population there. Now I want to touch on the H2 clade, which is particularly interesting for its very wide geographical range in the US, by far the broadest of the three lineages. You can see that it is all the way over here in Arkansas, probably in Oklahoma at this point, but it's also up here in New Jersey. So this suggested to us that this lineage might have been an early, if not the earliest, US introduction. We actually got a chance to look into this when the USDA sent us a sample of 2 legs from the first known Asian longhorn tick in the US, which was a larva found in Tyler County, West Virginia here, way back in 2010. And unfortunately, we were not able to squeeze a whole mitochondrial genome out of this sample, but there was enough DNA to confirm that it belonged to the H2 lineage. So for sure, we know this group was introduced to the US at least 15 years ago, which helps to explain some of the sequence variation we saw. The majority of US specimens we sequenced were, again, clonal or identical to one another. They're shown here in red. But there is this one sample from Tennessee that has a single mutation in the COX-3 region, not shared by any other ticks in the H2 dataset, which leads us to wonder whether the mutation arose after arrival in the US. That seems to be the case for these two ticks from Virginia, which share a unique tRNA mutation. Again, not found anywhere else in this clade, but the same mutation has arisen independently in several other lineages. So we know it's a polymorphic site, and believe it's most likely something that happened in these ticks after they arrived in the US, since they have had time to go through several generations, which is kind of exciting because it means we might be seeing the beginnings of genetic diversification in this new environment. Now, in terms of the source of this lineage, it's not abundantly clear, but the sample closest to the US genetically was from Japan. I want to clarify, a finding like this does not mean these ticks came from Japan. We did not have really good representation of overseas specimens in this H2 data set. We could not find any H2 data sets, H2 representatives from Korea or Australia, where we know this lineage does exist. So it is a very open case, but still it was interesting to see that most of the ticks from China formed a clade separate from the US. So not conclusive, but suggestive that this lineage likely didn't originate there either. And finally, we have the H3 lineage, which has moderate dispersal here in the US. This is a really busy clade. I know when we look at the US ticks, we see basically two things. So first, we have this clonal or identical group right here. These are all of the H3 ticks we have from the northeastern US. So New York, New Jersey, Delaware. They're all identical to each other. But then down here, we have these two samples from West Virginia. And to make it a little more clear what's going on, I'll switch to a cladogram view. So you can see that these US ticks are actually falling into two separate monophyletic clades. So the first is that clonal clade from the Northeast. And the second is, with very high bootstrap support, this clade down here that includes just ticks from West Virginia, Korea, and Japan. And because of this bifurcation between the clades and the way that West Virginia ticks are more closely related to Asian ticks and other US ticks, we know we're actually dealing with two separate introductions within H3. So that's not to say that these ticks were not part of the same introduction event, but they are certainly descended from two separate individuals brought to the US. As for the source of these individuals, it's not totally clear. We do again see the Korea and Japan connection in this one clade down here. I did, though, come into this analysis expecting to see some sort of connection with Australia or New Zealand, just knowing that these countries export livestock like sheep and cattle to the US. Sorry. But there actually was not a really strong support for that link with Oceania. So in this PCA, I know it's a lot to look at, but basically the US clades are in little, then in yellow. Over here you see the China specimens, and up here the Oceania specimens. And again, I know this is hard to see, but the US clades are both more closely related to specimens from South Korea and Japan. So again, we could really increase our confidence in this if we had more robust overseas representation. But as far as what we can take away from the data that we do have, first of all, we have learned that there were at least 4 separate parthenogenetic individuals introduced to the US. This does not necessarily mean there were 4 separate introduction events, but based on their kind of disparate distributions, it seems likely there were at least a few different introductions to different locations. And for all of these lineages, we did see the strongest genetic connections to populations in Far East Asia, specifically, like I said, Japan and South Korea. Again, I did not expect this, especially because these are considered a major cattle pest. I thought perhaps they arrived to the country on cattle. And the only countries, like I said, that have sent us cattle recently are Australia and New Zealand. So instead, we might have to consider these ticks could be coming into the country through different, less traditional routes. In the past, the import of companion animals, specifically from certain countries, has been pretty loosely regulated, not subject to the same inspection and quarantine requirements as commercial animal shipments. And when you consider it only takes one tick on a dog, for instance, to start a population here, it makes a lot of sense that they've made it. So the bottom line is that this tick can serve as a little bit of a wake-up call, letting us know that in this increasingly globalized world we're taking part in, we may be leaving gaps for exotic ectoparasite introduction, and that to prevent similar occurrences in the future, it might be a good idea to expand our vigilance beyond those commercial livestock shipments we're trying to protect to some of the less regulated hosts, like companion animals or exotic pets that we are regularly moving overseas. Okay, I'll try to move through this last part pretty quickly. Since we're moving on from the longhorn tick, I'm going to talk about the most abundant tick that I encountered in my New Jersey survey, and that was the Lone Star tick, Amphioma americanum. I recently moved to Wisconsin, and I'm really enjoying a summer without these guys. Those who live in their range know that they are extremely aggressive and they can reach really high numbers in some areas. And unfortunately, they are also pretty much on the move. Their range is expanding northward and westward, possibly representing a reclamation of former territory. In New Jersey, we see this expansion year over year as they intensify in the southern part of the state and move gradually further inland, where they used to be pretty much non-existent. Like I mentioned, they are very aggressive, which means that not only do they climb up on foliage and quests, like you'll see black-legged ticks doing, if they detect a host nearby, they will actively crawl towards it and just like approach you and run up your leg. They are very, very fast, and they will feed on a wide range of hosts, although they do have a preference for large animals like deer and cattle, and of course, humans. Their movement across New Jersey and across the country is concerning because this means new areas are increasingly exposed to conditions that they did not have to deal with in the past and may not be familiar with. A hot topic right now is, of course, alpha-gal syndrome, but these guys also transmit several pathogens, including a few viruses and several agents of ehrlichiosis, and that is what I'm going to focus on today. So ehrlichiosis actually refers to a few similar diseases caused by bacterial pathogens in the genus Ehrlichia. Basically, illness is a result of those bacteria infecting and destroying our white blood cells, though different species do target different types of white blood cells with slightly different manifestations. I hope you enjoy this AI-generated image that illustrates that happening. Very realistic. The resulting symptoms from all the Ehrlichia infections are very nonspecific, of course, and similar to those of a lot of other tick-borne diseases. So fever, aches, chills, etc. If you go to the doctor and you have a physician with the knowledge and resources to diagnose ehrlichiosis, this can be easily treated with antibiotics, the same ones that treat Lyme disease. But some cases of ehrlichiosis can be dangerous and even fatal if treatment is delayed or not administered. So the bottom line here is that fast and accurate diagnosis is crucial. But that can be a little bit messy because, like I mentioned, there are several bacterial species that cause ehrlichiosis in the US. The ones transmitted by Lone Star ticks are Ehrlichia chafiensis, Ehrlichia ewingii, and the more recently described and very poorly understood Panola Mountain Ehrlichia. And unfortunately, serology tests, which in the past have been the most common way we diagnose ehrlichiosis, can't actually distinguish between Ehrlichia chafiensis and Ehrlichia ewingii. And it's actually not clear whether they are able to detect Panola Mountain Ehrlichia at all. Which leaves us with very little information about the actual respective burden of each of these pathogens. What we can do, though, is to approximate this, we can use ticks as a proxy, screening them to find out what they're carrying and what people are most likely to come in contact with when they're bitten. We originally tried doing this with existing molecular assays, but we were having some issues with sensitivity, especially when testing for Panola Mountain Ehrlichia, and we were running through our DNA extracts pretty quick. So I started to look into the possibility of designing an assay that would allow us to accurately and efficiently screen ticks for all three of these pathogens at once. So this, of course, would be done with multiplex qPCR. You're probably familiar with this concept, but just in case, a refresher is that in qPCR, we start with a pair of primers that will amplify a region of DNA like traditional PCR. But unlike traditional PCR, we also include these fluorescent probes, which bind to that region between the primers. And when they're interrupted by DNA polymerization, they give off this specific fluorescent signal that our qPCR machine can detect and quantify throughout the reaction. So a really big advantage of this method is that we can include multiple probes here with different reporter dyes that will allow us to test for several different targets at once and even see if a tick is carrying more than one pathogen. So to apply that, we look to the 5' end of the 16S gene, partially because it's really commonly sequenced. So there's a lot of genetic data available. It also happens to be really ideal for an assay like this. So here in this alignment of the three sequences we're interested in, you can see that on either end, there are these two conserved regions. They're the same for all three pathogens, which means we can place these general primers that will amplify all three pathogens. And between them, there's this variable region, especially right in here. So we're able to create these species-specific probes that will theoretically detect only one pathogen each, emitting a different fluorescent signal to tell us which one or multiple are present. So once we are happy with this on paper, we moved on to validating it in the lab. Much of this was done with synthetic DNA constructs, representing the 16S region of each pathogen, although we did confirm everything using real tick DNA extracts. And I'll spare you too many of the details unless you would like to hear more about them. But basically, we confirmed everything worked well as individual singleplex assays, and then we combined all our reagents together into a multiplex. We tested to make sure the assay was sensitive and specific, so able to detect just a few copies of DNA if needed, but also not prone to amplifying anything outside of those 3 target species we're looking for. I'll mention we did test this assay out with synthetic Ehrlichia ruminantium, which is the species responsible for heartwater disease in cattle, because it is very closely related to Panola mountain Ehrlichia. And we confirmed that our assay does not detect it. Sorry. So once we were feeling confident in this assay, we could use it finally to answer our original question, which we would do by screening Lone Star ticks from across the state to assess Ehrlichia prevalence and distribution. These ticks we tested, they came from, of course, that statewide survey of forested sites that I covered. By the end of that survey, we had over 1,300 adult Lone Star ticks. You can see a male here if this video plays. Yeah, grabbing onto our drag. The map on this side shows our distribution of sites. The ones marked with circles, closed circles, are ones where we collected at least 10 adult Lone Stars over the 2 years. As you can see, these are concentrated in the southern part of the state, or the historical range. Those open circles, we had kind of intermediate numbers, and then we did not detect them at the X's. There are certainly more substantial sample sizes in the south, but you can see that they're generally slowly encroaching on the northern part of the state. So we brought these ticks back to the lab where we extracted their DNA using Dana's very helpful Kingfisher extraction robot. Then we use these extractions as template in our triplex qPCR assay run on a QuantStudio 5 thermocycler. And then the associated software, we could visualize any resulting amplification. I'll note that we did not have a super high CT for any of these samples, probably because these were adult ticks, but we did use a cutoff of 37 cycles anyway. So here we have a broad overview of our results. This summarizes the statewide totals for each pathogen, the totals over the 2 years here in this column. Some of these numbers were definitely not what we expected. In the past, New Jersey and quite a few other jurisdictions have reported all of their serological Ehrlichiosis positives, so the clinically diagnosed cases, as Ehrlichia chafiensis infections, in large part because this is the bacterial species that was used to develop these nonspecific tests. But we actually found a much higher prevalence of Ehrlichia ewingii in about 8.5% of our ticks versus only 2.7% of them carrying chafiensis, which seems to be a little bit reversed from what they're seeing in a lot of other areas in the country, particularly out west. And we did find Panola mountain in about 1.5% of our adult ticks. This seems to be quite consistent with other reports from the Lone Star range, which typically find this pathogen to stay around or under 2% infection prevalence. Now moving on to distribution. These maps indicate the sites where we found each pathogen with a circle for presence or an X for absence. So we expected Ehrlichia chafiensis and ewingii to be everywhere. They're well established, and they pretty much were. But it was interesting to see that Panola mountain is also really quite broadly distributed in New Jersey at this point. We fortunately did not see any individual sites with an alarming concentration of Panola mountain. The highest local prevalence we saw was a site with 3.4%. So again, it seems like this is a relatively rare pathogen, even in adult ticks, at least in our area. And we did find ticks co-infected with each potential combination of these 3 pathogens, though we did not see any carrying all 3 at once, which would have been kind of cool. We used chi-square test to compare the observed and expected rates of co-infection and didn't find any to be occurring at a significantly elevated rate, more than would be expected. However, you can see that there were slightly more Ehrlichia ewingii and Panola mountain co-infections than would be expected by chance alone. So that's interesting and something to keep an eye on in studies with larger sample sizes. So as far as what we can take away from this data, we were really interested to find ewingii to be so much more prevalent than chafiensis. It seems like the reverse is true in a lot of parts of the Lone Star range. We cannot say exactly how this translates to human or animal cases of ehrlichiosis, but it does point to the potential importance of this pathogen in our area, and it certainly highlights shortcomings in our current diagnostic capabilities. This is something the CDC is aware of. You might have heard there was a case definition change in 2024 that affects how we report ehrlichiosis. So unless there is molecular confirmation done directly on the pathogen, typically PCR, the species infecting will not be reported. So basically, serological tests will no longer be classified automatically as Ehrlichia chafiensis. Now quickly, just pertaining to Panola Mountain, we confirmed this pathogen is present, albeit relatively rare, throughout the Lone Star tick range in New Jersey. It's something to keep an eye on as we see these ticks continue to expand their range and abundance in northern New Jersey and beyond that. We do not know much at this point about exactly how pathogenic it can be, but given how frequently Lone Star ticks feed on people and dogs, it seems very likely that at least exposure is occurring. And we want to emphasize, of course, that Panola Mountain can co-infect ticks with both chafiensis and Ewingii, which just speculatively might lead to dangerous manifestations. Again, there's a lot we don't know about how it behaves in hosts, but hopefully that changes as molecular testing becomes more common. With that, I would like to thank everyone who made it possible, especially my PhD advisor, Dana Price, who was so generous with his time and knowledge, as well as my awesome committee members and lab mates. And again, I'm currently the Midwest Center of Excellence for Vector-Borne Disease, working under Dr. Susan Paskowitz and Lyric Bartholomew, and getting to experience more of the applied side of vector research, which I think my PhD work really prepared me for, with a kind of a foundational knowledge of ticks and their complexity and importance. Oh, yeah. I think you're muted, Zach.
There we go. Thank you, Zoe. While people formulate questions, I'm going to post the post-webinar question. And then just share the tick. And then please use the Q&A section of the Zoom portal to ask your questions. Or if you like to speak, just request it. I already have one question from Jillian Lepman. Did you compare rural versus urban forests for Ehrlichia?
We did, yeah. There actually wasn't any difference either in any Ehrlichia prevalence or in Lone Star tick abundance or density, which I was more confident in than the black-legged ticks, because even with the small transects, these ticks are so abundant that you can get an idea of how many there were. And yeah, I didn't see any difference at all, which kind of surprised me, because I would expect them both to be related to like the density of white-tailed deer. And I see the question about Ehrlichia reservoirs. So all three of those pathogens, the Ehrlichia pathogens I mentioned, are thought to be reservoired in white-tailed deer, but it's very possible, especially for Panola Mountain, which we know very little about, that there are other wildlife hosts that are important.
We have a question request for Timothy Fox. Go ahead. Well, Tim, I think you are still muted if you want to ask your question.
Okay. Hey, I wanted to just commend the speaker for a great presentation, and I like the speed of her presentation, and I can comprehend it good. Sometimes the speed gets in the way of my comprehension, but this was good, and glad to see that you're moving to Wisconsin, because I think there's a research hole in Wisconsin for tick-borne disease.
Well, yeah, thank you so much. I'm glad the pacing worked out. I felt like I was going too fast. And yeah, it's really exciting to get to experience the tick-borne disease landscape in a new place. Even though, you know, I had that love-hate relationship with Asian longhorn ticks and Lone Star ticks. I kind of miss them like a little bit.
Well, we do have them in Wisconsin.
The Lone Star ticks sometimes.
They're all the way up to Washington Island, the northern.
Oh wow!
Off the tip of Door County, and scattered across the state. Yeah.
Yeah, like sporadically. Yeah, I mean, I'm sure they're moving towards this area, but I hope it doesn't happen too quickly.
Mm-hmm. They like Washington Island because it's surrounded by Lake Michigan, which keeps it warmer.
Yeah, the coastal areas.
Mhm.
Thank you, Tim.
Thank you.
Any other questions? We have one more from Joellen. I am often concerned that results showing higher rates of Borrelia burgdorferi in urban forests might lead to calls to remove these forests. Have you experienced anything that might dispel my concern?
Might lead to calls to remove the forests. Is that what you said?
Correct.
I haven't experienced anything like that. I think if anything, um, it means we should be. I mean, a lot of these are like really high use. I think if anything, it means we should be putting up signs and just focusing on education so that people are kind of aware and vigilant when they're in these forests. I don't know specifically how much they could contribute to actual human tick-borne disease because we know a lot of exposure does happen in residential settings rather than parks where people might be a little bit more vigilant. Um, but yeah, I hope it doesn't lead to any calls to remove them. I don't think that's most likely going to happen. We love our forests in New Jersey.
Then another question from Keith Price. Did you say that you decontaminate / wash tick service before you qPCR? If
Yeah,
so,
sorry.
bleach used? If so, any concerned solution could enter tick and disrupt DTV RNA.
Ooh, so yeah, so we do surface sterilize. We do use a bleach ethanol wash and successive washes with water. That actually is something I was interested in. So a experience I had with DTV was that ticks collected in ethanol from a site where we had ticks that had DTV. They were all contaminated. I surface sterilized them and they were all contaminated with deer tick virus at one point, but like very, very high CTs. So you could like tell the difference between a true infection and ones that were contaminated, which made me think that the DTV was actually in the ethanol and possibly entering the ticks. So I started collecting them dry after that. But yeah, that is definitely a concern, especially with deer tick virus. We did pretty painstakingly surface sterilize them. So I believe it was a problem in the ethanol. Also, I tested the ethanol. I just remember directly, and it was positive for deer tick virus.
And then just to advertise the next presentation, we have Dr. Kendall Bromwiller from University of Southern Carolina presenting on some work she did surveying mosquito control in the Southeast. And then she'll be presenting next month, August 27th. And the registration link will be shared through the Basecamp next week. So sign up, and I will also be advertising in the Tick Academy. Thank you, Dr. Narvaez, for presenting to us today, and hope to see more of you all of you and more of you, more webinars in the Tick Academy. Thank you all.